All right, now moving on to how the cell membrane transports um, solutes from inside the cell and then outside the cell. There's a couple ways that this happens. Um, Specifically, when we're talking about the proteins and the way that that works, the first kind is facilitated diffusion. Um, and there's a couple of different proteins on the cell membrane that can help with facilitated diffusion. And the key here with facilitated diffusion is that there is only kinetic energy that's pushing the solutes through the cell membrane. Um, and that means that there's no energy that the cell itself needs to exert. So there's no ATP. And this also means that there's no, or that there is directionality. And specifically, the direction has to be from high to low solute concentration. And it's only one direction. You can only go one direction at a time. So if we see here with these couple examples of proteins that I have, this protein right here is what we would call a channel protein. And most channel proteins have this little gate thing right there. And there can be different stimulus that impacts when the gate opens. There's voltage gated channels. There's mechanically gated channels, and there's ligand gated channels, etc. These are the main ones. And so when there's a change in voltage, then the gate will open, or when there's mechanically changed thing, then the gate will open. If there's a ligand that attaches, the gate will open, etc. Um, the other kind of protein here that we have is just a carrier protein. Um, and this is where solute can come in, particle can come in and attach here, and the protein will change shape and let it come out uh, inside the cell or outside the cell, whichever way it's facing. Um, and this is called carrier mediated because it uses a carrier protein, and it's kind of like a revolving door where it's only open to one side at a time, and then it channels and shuffles the protein through. Now, there's also leak channels. We don't have to talk a lot about that right now, but those open randomly, and those are important in keeping a chemical gradient at equilibrium. But um, yeah, that's mostly it for facilitate diffusion. Important note here is that because we are using transport proteins they're in the cell membrane, right, and they're proteins along the side right here, because there can potentially be a limited number of these proteins. The rate of diffusion depends on how many proteins there are to transport the solutes, and it also depends on the concentration, where if there's more higher concentration of a certain particle in the cell than outside the cell, the direction will go this way, which is exactly what we would want, right? But the rate would also be higher if this difference in concentration gradient is higher, because then the likelihood here inside the cell, the likelihood of a certain protein hitting or a certain particle hitting a protein is a lot higher, and so more particles will shuffle through those proteins. Um, Rate can also depend here on the temperature as well, because as you increase the temperature, you increase the kinetic energy, which is what facilitated diffusion is based on. And all of these proteins also have very high specificity, where typically only one kind of a particle is allowed through, or only one kind of a molecule is allowed through. Now, when there are molecules that are very similar to each other, like glucose and galactose, there can be a situation here where they are competing, and if there's a lot of galactose in a cell, then that might overrun um, the proteins and then glucose won't be able to get in there if the lactose concentration is too high. So there is an effect here of competition. In competition, if there is a particle that's really very similar to the target particle, then there will be competition and a decreased rate for glucose. But typically, the specificity of most protein transport proteins are pretty specific, and they don't accept other things. Now, moving on to active transport. The key with active transport is that active transport requires ATP. And this is because the directionality of primary and secondary active transports is different than facilitated diffusion, where the gradient and the direction has to be, or I guess it doesn't have to be, but typically it is from low to high. And so instead of moving proteins from inside the cell to outside the cell when there's more inside the cell, active transport would move these particles outside the cell into the cell, even when that's not its natural concentration gradient. And because of this directionality, it requires the use of ATP. And this is kind of an outline here of of what a primary active transport protein would look like. They can be uniport, antiport, or symport, which means just determines the direction and how many particles it can shuttle. Uniport means that there's only one particle shuttling one way. Symport means that there's two particles that are traveling the same way. And antiport means that one particle is going one way and the other particle is going another way. And the way that this works is, and the example that I have here is the sodium potassium ATPase pump, where you have sodium here in the intracellular fluid. It comes and it attaches to the primary active to the pump, and that causes a conformational change, and it requires the use of a phosphate group right here, which is produced from ATP, and it causes a change which then expels the sodium, and because the sodium leaves, that causes another change where it allows it to accept potassium, and so potassium then attaches, and two potassium attach on the outside, and then that causes another conformational change, which causes the protein to dump these potassiums into the cell, and then a phosphate group comes back on from ATP in order to start this process all over. Um, and so, as we can see here, because it requires the use of transport proteins, the rate would also increase as the proteins increase, the amount of proteins increase. Uh, and it's not as affected by concentration gradients because it's pulling the particles from typically from low concentration to high concentration. There's also still very high specificity because it's a protein, where there's only certain particles that can enter. You can see that I illustrate that in my drawing here with the different shape of um, sodium versus potassium attachment sites. So we see that there. Now, the other kind of active transport is secondary active transport. And this is based off of primary active transport. It uses the gradient that was established from primary active transport to continue to move particles. So if we have sodium, we have a little bit of sodium inside the cell, not a ton, but there is a lot of sodium outside the cell. And this is because of the primary active transport, sodium potassium pump that made that happen. Now, secondary active transport uses this concentration gradient that sodium now has, and it uses it and it uses that potential energy to pull other particles against their concentration gradient. For example, um, and these can be secondary active transport can also be symport or antiport. And so what could happen is sodium wants to come into the cell, so it goes into the cell through this protein, and then it drags calcium with it. even though calcium's concentration gradient would want it to go that way. And that's because it's using the energy from sodium's concentration gradient to pull calcium with it. So it's still based on ATP, but it does not directly use ATP. The ATP from primary transport allows the concentration gradient of one particle to be established. Secondary active transport then uses that concentration gradient to pull another particle. This could also be the case where, say, you have a lot of glucose inside the cell. Um, then, or I'm sorry, here with the calcium, I was talking about that a little bit wrong. The Say there is a high amount of calcium inside the cell already. So I forgot to do all those extra calciums. Then the calcium doesn't want to come in. The concentration gradient should be going that way, and so it goes against its concentration gradient because sodium is pulling it along with it. Um, anyways, so for this other example, I doubt this is actually what happens in the body, but just as an illustration of antiport uh, movement, say that the cell wants the glucose out of the cell, then it can use the gradient from sodium comes in, and because of that energy that there is from the sodium coming in, it can also allow glucose to go out, even though it's against concentration gradient. So those would be examples. Antiport, antiport here, symport there. Now, osmolarity is also another very important principle. And really, osmolarity is just dealing with the movement of water, H2O. And water moves from just as particles want to move from high concentration to low concentration, we can think of it the same way here. And water wants to move from a high concentration, or sorry, a low concentration of solutes to a higher concentration of solutes. And that's because if you have few solutes out here, there's more water outside the cell. And so the water would want to rush into a place where there is less water. And that correlates with particles, where the less particles there are in the solution, we would call this outside hypotonic compared, or sorry, hypoosmotic compared to this inside solution. And this would be hyperosmotic. So that's how osmosis works. And osmosis is based on osmolarity rather than just basic molarity. And molarity is the amount of moles of a certain um, molecule. For example, if you have sodium chloride, moles just counts the amount of those molecules. And so 3 moles of NaCl is 3 moles of this entire thing. But what happens is in solution, that actually splits into 2 separate particles. And osmolarity and how water moves, water is based on particles and not molecules. Water doesn't care if the sodium chloride was together before. In solution, they're separate. And so osmolarity helps us determine the amount of particles. And so in this situation, if we assume that sodium chloride dissociates completely, a 3 molar solution would actually be a 6 osmolar solution. So osmolarity is based on the number of particles. Less particles um, in compared to a certain point, right? If there's less particles, that means it's hypoosmotic. If there's more particles, then it's hyperosmotic. If you have 2 solutions and they have the same amount, then that's isoosmotic. Now, the difference between osmolarity and tonicity is also an important thing, because just because something is um, Isoosmotic does not mean that it will be isotonic. Tonicity refers to the effect on the cell. And you have to take this into account after the movement of particles happens. For example, um, say we have a cell right here, and it's in solution, and there is a lot of glucose here. Now, and then there's also a few other sodium particles here. Sodium is non-permeable, which means that it can't move. Glucose is permeable, though. So what happens is the glucose out here, because there's more outside than inside, will want to come into the cell. And the problem is that that cell will actually metabolize the glucose. And so when it comes in, it gets deleted or it gets rid of it almost immediately. And so what happens is this glucose disappears from the outside solution and it doesn't appear inside the cell effectively. And that ends up causing the cell to actually be in a hypotonic solution because the glucose is gone and there's more particles inside the cell than outside the cell. And so the water would want to enter the cell. Um, and so in that situation, we can see how an isoosmotic solution, just because something has the same amount of particles, does not mean that it will be isotonic, which refers to the effect that it has on the cell. The cell would not necessarily stay the same size. And so this would be hypotonic, or I'm sorry, hypertonic, where cell would want to rush into the cell, where water would want to rush into the cell. And if it was the opposite, where there's more particles outside, then if there's more non-permeable particles outside than inside, then water would want to rush out, and that would be hypertonic. And so while osmolarity is concerned with the number of particles, tonicity is concerned with the amount of non-permeable particles.